Meiosis is a cell division process where a single cell divides twice to four cells that have had their genetic information, going from a diploid cell to haploid cells. Meiosis is used in the human body for the production of gametes. Similar to the interphase of mitosis, the cell grows and replicates its chromosomes by copying the cell DNA and sorting it into chromosomes. In this stage, the chromatin is loosely distributed, considered uncondensed. The two centrosomes are made from an original centrosome, which is remaining. In these centrosomes, we can see the centriole pair. In prophase 1, the longest stage in meiosis, the chromosomes from interphase begin to condense. These homologous chromosomes additionally start to loosely pair with each other. This happens so the chromosomes can match up at the various positions. Then the process of crossing over happens as the DNA gets broken and reconnected so homologs can exchange part of their DNA. The tetrads combine the sections that overlap from the synapses. The spindle microtubules pull the homologous chromosomes apart by attaching to the present kinetochores, which causes the nucleoli to disperse into two. Kinetochores attach to microtubules and homologs move towards the metaphase plate. During metaphase 1, chromosomes line up by homologous pairs. These pairs are arranged at the metaphase plate with each chromosome in the pair facing each pole. This forms tetrads. Both chromatids of one of the homologs attach to the kinetochore microtubules of one pole, and both chromatids of the other homolog attach to the microtubules of the opposite pole. Mendel's law of independent assortment is at play in this phase as it determines the genes each gamete will contain as to gain one of the two homologous pairs. Anaphase 1 begins as each pair of homologous chromosomes separates, which is caused as the proteins responsible for the cohesion of sister chromatids are broken down. Added by the spindle apparatus, the separated homologs move towards opposite poles. Cohesion between sister chromatids still occurs at the centromere, letting the sister chromatids move together towards the same pole. In this phase, Mendel's law of segregation is at work as each homologous chromosome pair is segregated with various versions of the gene. In telophase 1, each cell has a complete haploid set of chromosomes which were separated in anaphase. The chromosomes are made up of two non-identical pairs of chromatids, showing how meiosis randomizes genetic material. The chromosomes do not replicate in this stage, but just split. In cytokinesis, the cell splits into two between the cleavage poles. After the cell splits, prophase 2 occurs to ensure that a spindle apparatus is formed in each new cell. In the later stage of the phase, the chromosomes move towards the metaphase 2 plate. In metaphase 2, the non-genetically identical chromosomes realign as they are about to be pulled across the cell by the spindle fibers. The kinetochores attach to the microtubules and get pulled across the metaphase plate. During anaphase 2, the separating centromeres migrate along the spindle fibers in order to reach the sister chromatids, the opposite poles. The separation and movement of the centromeres is due to the shortening of the microtubules. Mendel's law of segregation can be seen through the process of anaphase 2 as the meiotic division within the homologous chromosome is segregated into two daughter nuclei with their own version of the gene. In telophase 2, the chromosomes arrive at opposite poles and begin to decondense. Once the chromosomes start to decondense, a nuclear membrane begins to form around the chromosomes. The process of cytokinesis begins when the chromosome completely splits into two new cells. The outcome of cytokinesis results in the final outcome of meiosis, which is four unique haploid daughter cells that each have a singular chromatin. However, at this point, the cells are not yet gametes. The process where the haploid cell turns into gametes is known as gametogenesis. Spermatogenesis produces four haploid sperm cells, while the process of oogenesis can produce a singular egg cell. The gametes produced during meiosis are all genetically distinct.